Assalamualaikum and hi, I greet to all lecturers. Before I start my presentation, I would like to introduce myself first. My name is Khairatul Farhana binti Marosi, and in this video, I would like to present the proposal of my final year project. First of all, the topic of my research is determination of the presence of antimicrobial resistant bacteria and the level of antibiotic residues in catfish and soil samples from catfish breeding pond Sungai Kesang, Melaka. My supervisor is Dr. Siti Shahra binti Zulfaqar and my co-supervisors are Dr. Muhammad Syam bin Uthman and Cik Nur Faizah binti Abu Bakar. Here are the contents of my proposal presentation, and now let's jump into the introduction. Aquaculture is one of the best solutions to meet up with the increasing demands of fish products. There is one important species that is produced from aquaculture, which is African catfish, or also known as Clarias cariopinus. This species is produced in many parts of regions in the world, including Malaysia. But due to some diseases, fish farmers are forced to use antibiotics mostly for treatment and to prevent infection. However, the misuse of antibiotics in aquaculture and other animal farming had caused the emergence of antibiotic resistance issues. Apart from antibiotic resistance issue, antibiotics can reside in environments such as soil, crops, and coastal surface water, and it is called as antibiotic residues. African catfish, or also known as Clarias cariopinus. This species was introduced into Malaysia from aquaculture sector in Thailand between 1986 to 1989. And this is the highest finfish species production in Malaysia after the domination of seaweed in aquaculture. In this research, Sungai Kesang that is located between southern state of Melaka and the state of Johor is chosen. As you can see in this map, the yellow dashed line is the border of Melaka and Johor, and the white line is Sungai Kesang. Sungai Kesang, with approximately 35 kilometers long, flow into Cincin water treatment plant, and one of the activities that contributes to contamination of Sungai Kesang is catfish farming. According to Thorsten Christian et al. 2003, there are two divisions of antibiotics used in veterinary. The first one is antibiotic for therapeutic use with animal for infection prevention, and the second one is to accelerate growth through feeding. In the last 2 to 3 decades, there are increasing growth of fish culturing industry that unfortunately resulted in harmful environment effect, and it caused the spread of antimicrobial resistance that has become a global public health concern. This is because the health risk caused by antimicrobial resistance is not limited neither among human health, animal health, nor the environment, as it can be transferred between these three media and it can remain in the host for a long time. The use of antibiotics can also result in deposition of residues in environment and animal tissue, and there are possible routes of entry into environment such as disposal of unused drugs, excretion of waste products, and incidental release of products from spills or discharge. The antibiotics reside in fish tissue could be systemically toxic to the consumer, giving adverse effects towards human body. Here are 3 examples of antibiotics and the adverse health effects. The first one is tetracycline. It can cause superinfection. The second one is penicillin. It can cause acute allergic reaction and spread of methicillin-resistant organisms. And the last one is sulfonamide. It can cause thyroid, adenoma, and hyperplasia. In order to conduct this research, there are several supportive reasons that are associated with this issue. The main problem is that the massive use of antibiotics in fish industry has become a common practice among fish farmers. Antibiotics are stable compound and they are not broken down easily in body but will remain active after long being excreted. And they also have high affinity to soil particulate matter and can have half-lives of more than 300 days. This research is aimed to know the safety level of the catfish in terms of presence of antimicrobial resistant bacteria and the antibiotic residues in it. The effluent from catfish breeding ponds might also give impact to the environment near it, such as soil and sediment. Next, my research question is there any contamination of antimicrobial resistant bacteria and antibiotic residues in catfish and soil samples from catfish breeding pond Sungai Kesang, Melaka? The general objective of this research is to determine the presence of antimicrobial resistant bacteria and the level of antibiotic residues in catfish and soil samples from selected catfish breeding pond Sungai Kesang, Melaka. For my specific objectives, the first one is to determine the level of bacterial contamination. The second one is to determine the presence of antimicrobial resistant bacteria. The third one is to determine the level of antibiotic residues. And the last one is to compare the level of bacterial contamination between different sampling points. My research hypothesis is there is a contamination of antimicrobial resistant bacteria and antibiotic residues in catfish and soil at catfish breeding pond Sungai Kesang, Melaka. Before I explain on the methodologies, now let's go through the research flowchart in short. First of all, I will do the sampling of catfish and soil at Sungai Kesang, and then there are two parameters that will be tested. The first one is microbial contamination and the second one is antibiotic residues contamination. For microbial contamination, first of all, the sample of catfish and soil will be prepared, and then the bacteria from the samples will be isolated for antibiotic susceptibility tests through this diffusion method. And last but not least, of course, data analysis. Meanwhile, for antibiotic residues contamination, the sample will also be prepared to be run in high-performance liquid chromatography (HPLC) system in order to determine the presence of antibiotic residues. For research methodology, these are the sampling locations of catfish breeding ponds. The total of 9 catfishes will be collected from S3, while for soil, sample will be collected from S1, S2 and S3, S1 composite, and also from S4. Now for the method of microbial contamination. First is the sample preparation. It involves homogenization with peptone water and cereal dilution, and then bacterial isolation, incubation for 24 hours, enumeration, colony differentiation, subculture, and antibiotic susceptibility test. For microbial contamination of soil, the steps are same as catfish except for the sample preparation. There will be drying, crushing, and sieving, and the cereal dilution is with the ionized water. After done with all the microbial contamination procedures, the antibiotic susceptibility test will be conducted, and this diffusion method will be used. So basically, in this part, the bacteria from the previous procedure, the bacteria will be placed on Mueller-Hinton agar, and then antibiotic disc will be placed firmly on the agar plate as shown in figure 6. And last but not least, the plate will be incubated for 18 hours at 37 degrees Celsius. Now let's move on to antibiotic residues contamination. Basically, any samples that will be run into HPLC system must be in liquid form. So for catfish and soil sample, there are several methods can be used to turn them into liquid form. And for catfish, the extraction method that I choose is solid phase extraction. First of all is sample preparation with standard solution and hexane dichloromethane into 10 grams of muscle tissue, and then homogenization, centrifugation, addition of sodium chloride into supernatant, second centrifugation, process of foaming, cool down, and again centrifugation, and last but not least, the sample will be loaded into C8 column for column cleanup. In column cleanup, it involves C8 column activation with methanol, water, and ethylene diamine tetraacetic acid in phosphate buffer. And then the column will be washed with 4% acetone in H2O and will be eluted with water in acetone. After that, the process of evaporation to dryness, and last but not least, it will be dissolved in 150 microliter of mobile phase. The extraction method that I use for soil sample is ultrasonic assisted extraction. First, the filter paper will be placed at end of glass column, and 1 gram of prepared sample is added. The glass column will be closed with 1 way stopcocks, and 8 ml of magnesium nitrate containing 4% of ammonia is added. The column is then placed in tube rack and immersed in ultrasonic water bath. Extract the sample for 30 minutes before it is placed in vacuum manifold. In HPLC system, the injection volume is 100 microliter. And in linear gradient elution, the solutions that will be used is diluted formic acid and acetonitrile in 2 different compositions. And at last, the antibiotics in the sample will be monitored by UV detector. Here are 6 antibiotics that will be tested in this research. They are oxytetracycline, sulfamethoxazole, ciprofloxacin, trimethoprim, erythromycin, and ampicillin. For the first, second, and third objectives, the statistical analysis that I use is descriptive analysis. And for last objective, I will be using one-way ANOVA. This is the Gantt chart of my plan during this semester. And inshallah, if the coronavirus issue is settled down, we will start sampling in April. Before I end my presentation, I am very thankful for the financial support through this research grant and also help from my supervisor and co-supervisors. Here are my references, and I think that's all from me. Thank you and Assalamualaikum.